part 3 of chapter 5. This will be the final video. So last time we talked about how carbohydrates in the form of glucose can be broken down through glycolysis, then the intermediate step to take pyruvate to acetyl-CoA, and then into the Krebs cycle, and then the electron transport chain. And now we're going to just briefly look at how some amino acids can also go through that whole series. Other amino acids can be transformed and feed into the intermediate step as pyruvate, and other foods like fats can be broken into fatty acid and glycerol. The glycerol can go through the whole series. The fatty acids can join in during the Krebs cycle, and then yet still other amino acids go through the Krebs cycle. And so this is what it looks like overall. Of course, to this image you would want to add your alternative pathway, the pentose phosphate pathway. But besides that, this really gives you the entire overview of how all the different nutrient sources can be utilized to provide ATP. Another way to look at it is this diagram, right? So this is exactly what we just looked at, flipped on its side. So carbohydrates in the form of sugars go through glycolysis, form pyruvic acid, get made into acetyl-CoA, go through the Krebs cycle, and then go into the electron transport chain. The nice thing about this diagram is that it actually says acetyl-CoA, so you remember that that has to happen before the Krebs cycle, and it also shows you the removal of CO2 here, and it shows you the final electron acceptor oxygen and producing water. So it reminds you of a few things. So you could even take this and add on a few extra little things for yourself when you study. This is showing you how when you eat proteins, it'll be broken into amino acids. Some of those amino acids go here, here, and here. Lipids get broken down into glycerol and fatty acids. Glycerol can go into glycolysis. It doesn't start at the very beginning, but that's fine. It goes into glycolysis. And then fatty acids go into the Krebs cycle, okay? So I'm not going to ask you which amino acids, right, because I'm not going over that, but I would like you to know that proteins are broken into amino acids, and different amino acids can feed into different locations of respiration. Lipids are broken into glycerol, fatty acids. Glycerol goes into glycolysis. Fatty acids need to go to acetyl-CoA and then Krebs cycle and etc. So lipids are actually considered the highest energy food source that we can eat. That doesn't mean healthy, okay? But they are per gram the highest energy yield. Okay, so now looking at the biosynthesis. So instead of here, we're talking about catabolism, right? Breaking down to get energy. Now we're briefly looking at anabolic reactions to build, put together. So if we need building blocks, we can take some of those building blocks out of glycolysis and make things like glycogen and peptidoglycan. Okay, it does require energy to use those building blocks and make a product. If we need to make lipids, we can also take from glycolysis and the intermediate step and make glycerol and fatty acids. To build fats from sugars is the idea, right? Because you're taking glycerol from glycolysis, you're taking pyruvic acid to make fatty acids, okay? So acetyl-CoA can be transformed into lipids, right? Acetyl-CoA is you took pyruvic acid, you removed a carboxyl group, you added the coenzyme, and it's acetyl-CoA, and that can be transformed into fatty acids. We can also use this, it's not shown here, but we can make cholesterol, we could make triglycerides, or we can make just simple fatty acids. So whatever we need, whatever type of lipids we need, okay? We do call this lipogenesis, right, to make lipids. And of course, we can do the same thing with amino acids. So just like we saw being able to feed amino acids into catabolism, we can take them back out. And so we can take things from the pentose phosphate pathway, Krebs cycle, or even the alternative pathway that some organisms have because they don't do this. So one note is that this is not saying that every organism does all these things, because most organisms will only have the top two, right? Most organisms don't have this alternative pathway. And so this is putting it all together, both catabolism and anabolism, right? Catabolic and anabolic reactions. And you have fermentation shown here. So let's go ahead and talk about fermentation now. So fermentation, you start out as you always do with the glycolysis and forming pyruvic acid, but you are not able to do anything past that. You're stuck with this pyruvic acid and you're stuck with this NADH, and you don't have the Krebs cycle or the electron transport chain. It's temporarily shut down because of not having oxygen. Okay, so pyruvic acid's slightly toxic. We need to get rid of it and put it into a safer molecule. Okay, and we need to be able to replenish the NAD. We need to oxidize NADH to become NAD plus, right? So NADH is the reduced form, NAD plus is the oxidized form. We need more NADH if we want glycolysis to happen. Why do we want glycolysis to keep happening? Because during fermentation, this is the only mechanism to get ATP at all. And yeah, it's puny. It's only 2 ATP, okay? So this is, in a way, it's wasteful, right? Because you have this molecule, glucose, that has the potential to give you a lot of ATP, and you're only going to get 2. But it's necessary. And for some organisms, this is all they do, right? Some bacteria, this is what they do all day long, and they do it very, very quickly. It's shorter pathway than any of the other things we looked at, and it's fine. As long as there's plenty of glucose around, this is fine. For us, we use enormous amounts of energy. We're a huge organism, and so what we do is that we will actually turn pyruvic acid into lactic acid, right? That's the burn you feel when you work out real hard. And then we will take that and turn it back into pyruvic acid once oxygen's available and finish what we started and get the rest of the energy out of those molecules. Pretty neat, right? So different bacteria, different organisms will take pyruvic acid and make it into different products. But again, remember the key reason that they do this, pyruvic acid's not safe. We need to put it into a safer molecule, and we need to replenish the NAD+. That's the biggest reason, so that glycolysis can keep happening. And so here I've summarized the key facts for fermentation for you. All right, so you're still doing redox reactions. You're still doing glycolysis to break glucose into 2 pyruvic acid molecules, and you are still getting to a net of 2 ATP per glucose. The key reason for doing fermentation is to replenish your NAD+ so that glycolysis can repeat. This is happening when you don't have oxygen, or for some bacteria that are very simple, they don't have any of the enzymes to do all the other pathways we've looked at. This is all that they have. Of course, this is not using Krebs cycle. This is not using ETC. Okay, so fermentation is not cell respiration. Okay, And then you're using an organic molecule synthesized in the cell, which will be the waste product. And this is the final electron acceptor. And so you might be saying, what? Final electron acceptor? We don't have an ETC. What are you talking about? Well, we still have to do a redox reaction to replenish NAD+, right? And so you need something to take this electron, right, from NADH to become NAD+. And this looks different for different organisms. The two main ways to do this are alcohol fermentation and lactic acid fermentation. I already talked about lactic acid fermentation, and you're familiar with it because of your muscles. Alcohol fermentation you're going to see in lab. And so I'm just going to give you an idea overall that there are a lot of different products of fermentation, especially when we're talking about bacteria. I'm not going to test you on these bacterial names and what their products are, but for lab, you'll need to know that you're using yeast, which is Saccharomyces. So you should note that this right here is a yeast, not bacteria, and it is used to produce ethanol, and that's what you'll be seeing in the lab. Other bacteria, other organisms, such as different types of bacteria, can make things like acetone, fingernail polish remover, you might be familiar with, isopropanol or rubbing alcohol, right? Acetic acid, vinegar, and then we have the lactic acid that I talked about, which can then also be used to make some cheeses, yogurt, soy sauces, and then we also have a different organism that makes propionic acid, which can be used to make Swiss cheese. These organisms, rather, can be used to make Swiss cheese. Okay, so I think that's everything I want to talk to you about for fermentation. Now I want to briefly talk to you about some biochemical tests. Okay, so we will make different types of media, and they will contain specific ingredients that will allow us to test whether or not the bacteria has the ability to do something particular. So you've seen this already with the SSA agar, a rather complex media. Not all of our media are going to have so many different things occurring simultaneously. Sometimes it's just one single sugar. Can the bacteria utilize a different sugar besides glucose? And that's a common test, okay? And so by doing different biochemical tests, you can identify what bacteria you have, okay? You typically start out with a gram stain, so you at least know if you're dealing with gram positive, gram negative, acid fast, and then you can do these different chemical tests, okay? So biochemical tests are looking specifically, can the bacteria do this particular thing? Can it utilize this particular ingredient that we're providing it with? And whether or not it can do that will depend on whether or not it has the enzyme or set of enzymes that are required for that biochemical process. And so that's why they're called biochemical tests. Some examples are the urease test, dehydrogenase test, decarboxylase, or decarboxylation. We typically call it decarboxylase. Different fermentation products and different fermentation sugars. Oxidation tests, hydrogen sulfide tests, right? So there's a lot of different biochemical tests, and specific species or groups of bacteria will test particular ways, and this will help us. And so we use this in conjunction with a dichotomous key. So this is showing if you did a gram stain and it was variable, and then you did an acid fast stain and it was positive, you know you have an acid fast mycobacteria, right? It has mycolic acid. And you want to know, is it rapid growing or slow growing? If it's slow growing, you're going to perform the urease test. And if it's positive for being able to use urea, then you will see if it can also use a nitrate. And if it can, then you know that you have the tuberculosis species. If it cannot, you have the bovis species. Back up here with the urease test, if it couldn't use urease, if you're here on the negative side, then you know you have a Mycobacterium avium species. So that's how we use biochemical tests, and also how we use dichotomous keys to identify a bacteria. You do not need to memorize these particular facts here. This is just a demonstration of how to utilize these tools. Okay, so this is what a fermentation test looks like. This is an uninoculated media. It's red. It has the phenol red indicator, pH indicator, and all of these tubes start out just like this tube with the red color, and the only sugar that it contains is mannitol. And so we wanted to see, can the bacteria use mannitol? And so in tube B, we've put Staphylococcus epidermidis, and you can see that it was able to use the protein and not the sugar. So it's actually negative for fermentation, and it's alive, and it's producing an alkaline product from using protein, and that's why it turned orange. On the other hand, we have Staphylococcus aureus, which grew and it ate the mannitol, and the fermentation product is acidic, and that turned the pH indicator yellow, and that is positive for fermentation of mannitol. Here we have, not only is it positive for fermentation of mannitol, it also produced a gas, which was caught in an inverted small tube called a Durham tube. And this is actually E. coli. So E. coli was able to produce gas and acid from fermentation of mannitol and turned very yellow. So positive, positive, negative, and uninoculated. No bacteria. Okay. All right, next test, urease test. You're supplying urea, and you want to see, can the bacteria use urea as its only nitrogen source? If it has the enzyme urease, then it will, and the control is this peach color, and a positive is this pink color. And if pink is anywhere in the tube, so if it's only along the surface, that's a positive. If it's only inside, that's a positive. Any pink in the tube will be positive. Because some organisms, you'll just grow along the surface because they need oxygen, and some, when you stab, will only grow inside because they can't handle oxygen. So okay, so that's another biochemical test. We have other biochemical tests to test for protein, different proteins, different amino acids. You don't need to memorize this, but just the concept, right? And then, so detecting amino acid using particular amino acids shown here. And then the hydrogen sulfide test. I know you saw hydrogen sulfide already on the SSA auger, and this is another way to do it. This is a sugar peptone iron sugar auger, and you stab it, and then you can see that in the stab, and I know it's not always easy to tell in these images, but you do have some growth here of bacteria. So they grew, but they did not utilize, they did not produce hydrogen sulfide. Whereas in this stab, you see a lot of hydrogen sulfide was produced. Okay. So that is it. I put these check your understanding questions at the end for you. You should also use your textbook. They have questions at the end of each section. They have questions at the end of the chapter. All right, good job. That's it.